Looks like I'm live. Waiting for Jessica to arrive. She should be here momentarily. Welcome, everybody, to the Last Boomer podcast. It's been a while. This is my December one. Ah,
Oops.
hey, you know what's funny? I was going to play a song that I never knew the name of. And it was funny. It came up on my screen. And on the bottom of my screen, it said Jessica. And I thought to myself, why didn't I use that song? Why did I use the surfing song? Because I know you like surfing. So I was like, I'll pick a surfing song. I was going to do Wipeout, but then I did that Miser Lou. I don't know if you ever knew the name of that song. But
There's a
then,
famous song in Israel called Jessica that I hate because every time somebody meets me, they sing it to me, and I'm like, yeah, I haven't heard that before.
it's so funny. The one that I'm talking about, Jessica, I believe is instrumental. I don't think there's any.
Oh, nice.
So are you familiar with the song from the Allman Brothers?
Nope.
You're
I don't
kidding me.
think. I don't
Oh,
think.
you know what? Even though you're 10 years younger than I am or more, you will know this song. It's a very famous song by the Allman Brothers. I never knew the name of it until today, moments ago, when it came up on my computer. And on the bottom, it said Jessica. And I'm like, what does that mean? Jessica, why does it say Jessica? And then I Googled or whatever in YouTube. I said Allman Brothers Jessica, and it came up. That's the name of the song that I've been listening to for 50 plus years. I never knew the name of. And you would know the song. It's that famous. I'm going to ask you if we can change the topic a little bit, a lot.
Of
we can talk
course.
about we can talk about the DNA stuff, but listening to you and Kevin on
Marty, well, TFTC.
TFTC, that was great. You guys did a great. You nailed so many different things where people have questions as to what happened in society. Like, why don't more people know this? Why don't more people know that? And you cover like really why. It's because the journals are all captured.
Good.
So it was fantastic. No, I
It's
really,
true.
yeah,
It's like the key to censorship. Like people don't understand how that is the heartthrob of what happened. Maybe we should talk about this when we talk.
well, we are live. I mean, all this is being captured. We're live
Oh, okay.
here.
Well,
Yeah.
great. So I don't know if you know what's going on in Australia, but like they had this recent, I won't make any, personal comments on what I know the deal is, but they had this recent tragedy. They've since shut down New Year's Eve, which sounds a lot like COVID to me. And they're now doing this retrieval of guns from anyone who owns a gun, like right now. And it's like,
They don't want you to
oh,
protect yourself. You can't
yeah,
protect.
and the point that I wanted to make is that they're censoring you from being able to speak your mind about what the real goddamn problem is. And everybody knows what the problem is, but you're not allowed to talk about it, which is exactly the playbook that the UK has been using. So it's like everybody knows what's going on, but like we're not allowed. Censorship is the fulcrum of this frickin' propaganda shit that's pushing all this forward.
Yeah.
Like none of it would work without censorship as the fulcrum.
Yes. And it's been a long time coming where all the pieces were put into place. Censorship in the journals, censorship in the board certification. So you've got the overarching NGO in the United States is the Federation of State Medical Boards, and they basically control through messages that they send out somehow. It's a RICO organization. So the top part of that RICO organization is the FSMB, and below that you've got the American Board of Internal Medicine, American Board of Pediatrics, American Board of Family Medicine, and so forth. There's probably 100 different boards. That whole censorship industrial complex, as RFK Jr. talks about, has been in place a long time. And they just executed very well during COVID. And it bleeds into, of course, the journals. You're right. It's been in place for a long time. Could not have done it without capture. If it's okay with you, let's start out with introductions. So I'm the last boomer. This is my podcast. I say I'm the last boomer because I was born in the second half of 1964. And that's the last of the boomer birth years. So, Jessica, can you introduce yourself, your degrees, your experience, and before you do it, a lot of people know me as an anti-academic, and here I am talking to a pure academic. So go ahead, let's go, and you can talk about how we know each other and on the Kirsch calls or whatever you want to say.
It's so funny that I'm considered a pure academic. Wow. I've been through the whole washing machine of it all. My whole career, and for people just listening, I put that in air quotes, has been in academia. I'm basically just a full-time student. I've accumulated 5 post-secondary degrees. I'm just basically someone who likes learning about everything. Sometimes I feel like it's more of a deficit than an asset, that I'm curious about absolutely everything because I can't know everything.
Go deep though, Jessica. So opposite of me, where I stay high level, and if it gets too deep in the weeds, I go off to another thing. I want to stay high level from a systems point of view. But I want the crowd to know you're very specific with molecular biology, also with statistics and computations, and finding true evidence at the micro level, at the molecular and cellular levels.
Mhm.
Whereas I'm like really high up in the data. So I just want people to realize that it's what you do is esoteric. You're very specialized, very knowledgeable, very professional, and that's important for people to know. Sorry to jump in. You're talking about.
No, no, I appreciate that. And it's a pro and a con being esoteric. It's wild. I mean, Kevin McKernan and I have, we're working together all the time now. You know that very well. This guy in my eyes is like probably one of the smartest human beings alive, and I really mean that. And it's not because he's got this pathway that I've taken. It's actually very different. He went straight into doing what he had a tendency toward and what he was good at, which is what everybody should do, by the way. And he just like milked that for all it was worth. He's still doing that. He's a genomics expert. He knows more on this subject matter than anybody I've ever met and probably ever will. So, in that regard, it is a niche environment, but like we're what we're talking about really does involve every human being on the planet right now, because like a huge proportion of the human population, it's crazy that this happened. I still can't get over it. Was injected with at least one of these nucleoside modified mRNA shots. And what people might not know, which is what we're uncovering through, sequencing data and quantitative assays, is that there is DNA in these shots, and it's not a small amount of DNA. And it's in contrast, this finding is in contrast to what all the regulators and the manufacturers have been touting for years, that they still maintain on their websites, that there's no possibility, no remote possibility of your DNA being affected, quote unquote, by these shots because they're mRNA based. But guess what? If there's contaminating or residual DNA from the plasmid via production in the vials and it's being injected into you,
Yeah, it's
you
just
can't rule out that possibility.
for the audience. She's basically saying that the Fed and the pharma all lied in that they said it can't integrate into the human genome. What I'm trying to say is they lied. They said it can't get there, can't integrate. And yet we now have some proof from some labs that it does. And what is that? Is it the Netherlands or Denmark, or where's that from?
Well, there are two things I want to say about that. One, the implications of disturbing the genome in specific ways, like genomic instabilization, I don't know if that's a word,
Destabilization.
promoting genomic instability, um, isn't necessary to induce cancer pathways, but that would be what you would be scared of if you messed up your genome by throwing in a bunch of foreign DNA. All you need to do is introduce foreign DNA into the cytosol, the cytoplasm, the guts of a cell, in order to induce cancer pathways. So we don't even need integration. Integration is another problem that we really have to be concerned about from a germline, or stem cell point of view. Because if that happens in certain people, then that's a generational problem then. And, this is all speculative for now, but the thing is, in biology, and when you're messing with biology, you have to be cautious. The precautionary principle was basically like thrown out into the garbage on the subject matter of these shots. And all of the things that the regulators knew or should have known, i.e., the lipid nanoparticles biodistribute, they can go everywhere. The ovaries, the testes, this is documented.
Lie number one.
Frickin' 10 years ago.
Yep.
Um, yeah, and what happens when those LNPs dump their cargo? Their cargo being nucleoside modified mRNA that gets translated into spike proteins. Those cells are targeted for destruction by cytotoxic T lymphocytes and DNA, which can induce cancer pathways. It's mind blowing.
Let me just interrupt. I'm going to keep interrupting you if you don't mind.
No, keep interrupting me.
I'm going to try to break it down for the regular audience, and
I love it.
I might not do a good job.
No, I love it. This is what we need.
You use the word speculative. So people listening who are on the fence are immediately going to dismiss what comes after. Much of what you just said is not speculative. We do have conclusive evidence that certain genetic sequences from the vaccine got into human, say, cancer tumors. Is that correct, or am I off base?
Yes, that is a fact.
Okay.
But we need to reproduce that on a larger scale in order to conclusively say this is a problem. We are, what's that word? Conservative as real scientists.
Yes,
we're skeptical. We use the precautionary principle. We don't just come out and fearmonger like the narrativers are doing. We want hard evidence. And that requires reproducibility. And we know that there's a problem here, but we can't come out and definitively say exactly what the problem is until we have reproduced evidence of that finding or that hypothesis. So we're doing what you should do in science. We're gathering evidence, we're reproducing the results. After forming a hypothesis, we're proving theories, and then we're presenting conclusions from our studies. And that, that gets further reproduced. And the more momentum that gains, and this is key because all of this is being censored, the higher the chances that people are actually going to hear that there's a problem and that assays are going to be produced in order to help people detect whether or not their cancers are full of whatever, SV40 or whatever. And that might lead subsequently to solutions. And the very first part of the solution is to stop injecting people with this platform because it's notoriously bad and everybody knows it's bad.
Absolute poison, just absolute poison. And again, I want to jump in just for the audience. While Jessica's talking about maybe one thing, cancers and integration, that's just the tip of the iceberg. There's so many things wrong with it. And it's not just the SV40, which is, by the way, for the audience, again, it's a plasmid leftover from manufacturing that was supposed to have been taken out, but they got the product authorized under emergency use authorization for process one, which included the process step of removing what was left over and bad after manufacturing. And then they went to mass manufacture for 1 billion doses shipped out to the public, skipping the step of purifying or cleaning out or making the product not have the bad stuff in it. And so it's left in it and it causes cancer. That's just one thing. And we'll get into the other stuff later.
On that, just so I can piggyback, absolutely right. But just so that people know, the reason why the DNA actually got left behind for packaging in these lipid nanoparticles, and we know that happened. I don't give a shit if it's published in Nature. You can read the Ox paper. Anybody who knows anything about genomics or sequencing or writing good methodologies is going to find not only holes, but gaps in their methodologies. Anyway, that's all I have to say in that. This has been found in labs all over the world. You know, we're not conflicted, we're independent. All of us have been attacked independently, but somehow we've all managed to publish our results. It got in there because during the manufacturing process, synthesis process, which was the process two, which involved plasmids and E. coli bacteria, part of the plasmid, which is just a circular DNA, if you guys don't know, that has a little spike gene that is uptaken into bacteria. And every time the bacteria double, which is every 20 minutes, pretty much, I'm rounding the edges here. They take up this plasmid and they carry that DNA, which includes that spike gene with them. You break up the bacteria after a while, and then you linearize, which means you take the circular DNA out of its linear form to make it a line. And in that line, you have one little spike gene. And then you throw some shit in it and you do an in vitro transcription reaction with a T7 polymerase. So what that means is that you're going to be producing, with the addition of N1 methyl pseudouridines, the nucleoside modified mRNA from that DNA transcript. Okay. So what you're going to have.
Before you go on, I'm going to translate a lot of that. Okay.
Okay.
I make beer. I made beer for 30 years. Okay.
This is good.
You put a bunch of maltose in a bucket with some water, you cook it up on a stove,
I
and you
love
throw yeast in, and you hope that no bacteria got in there because you want the yeast to eat the sugars and produce alcohol and carbon dioxide rather than the bacteria producing bacteria poo. You don't want bacteria poo in your beer. All right. So it's a race. So the manufacturing process that she's talking about is a race. Well, it's not a race because they're purposely purifying stuff, but they're growing the product, okay, using the E. coli bacteria. And then you have to purify it. In beer, you let the yeast settle to the bottom when they go dormant, and you only filter off the beer that's sitting on top of all the yeast. And there's a little bit of yeast that gets left over, but you're not drinking a pile of yeast, you're drinking beer. So the point is, it's just she's talking about a manufacturing process. And although it sounds really technical, you make the soup, right? You make this big soup, and there's only certain stuff you want from the soup. You don't want the whole soup, right? You don't want the chicken bones and you don't want the cartilage. You just want the chicken stock and the soup. So that's what she's talking about. Go
That's
ahead. So,
beautiful, John. Thank you. Oh man, this is such synergy. Okay, exactly. So what happened in the broth? Let's do the soup analogy. So in the broth, you have all the bones and the guts and the cartilage and then you have this gorgeous bone broth, which is supposed to be that nucleoside modified mRNA that's pure. But you have both DNA strands and mRNA strands. And what happens because they added these N1 methyl pseudouridines to the mRNA?
which is not human. It's not human. Okay. So for the audience, right?
It doesn't come from nature. That's
Right.
true.
Thank you.
So they have a higher melting temperature and they're very sticky. So what happens is, because of reasons I'm not going to describe here, those two guys come together with higher affinity than other things. I'll just put it like that. So they basically go boom. And once they get stuck together, this is a DNA mRNA hybrid. And this does happen in nature, by the way, in your cells. And we have specific enzymes called RNase H that degrade these in our cells. But once this happens, it's very hard to pull apart because the melting temperature for these N1 methyl pseudo is very high. The main thing that people need to understand is that the enzyme that they use to break up the DNA in order to clarify the broth at the end of the process didn't clarify the broth and left behind all sorts of eyeballs and anuses in the form of DNA RNA hybrids. This DNase 1, it's so called, that they used can't rip apart these guys. They don't act on RNA or DNA independently. So that's why the DNA got into these lipid nanoparticles. But the weird part is that the EMA released in a document in, I think it was March
E-
2021.
EMA is
The European Medicines Agency, this
okay.
is
It's like our
regulator in
FDA.
Europe,
It's like the United States
it's
FDA.
European FDA,
Okay.
and they're a big deal in all of this regulation shit. They put out a document that detailed that they knew that there was residual DNA at a certain level, but what they claimed was that it wasn't something to worry about. And that's wrong on two fronts. Because number one, the DNA threshold for allowable, use in biologicals, for example, is 10 nanograms per dose, but that's based on naked DNA, which has a 10-minute half-life. We don't have a determined threshold for the amount of lipid nanoparticle
But
encapsulated DNA that's
yeah,
safe.
let me break
And
that down.
one more thing, we
Okay.
definitely don't have one that's safe for SV40 enhancer promoters, which are nuclear targeting sequences, for example.
Okay, so naked DNA being stuff, you all eat hamburgers. You get DNA from cows and steer in your bodies, right? And some of it gets into your bloodstream. When she says naked DNA, it's not encapsulated with an LNP, lipid nanoparticle. Okay. The important thing is it's not the naked DNA or just the DNA floating in your bloodstream that's going to be broken down by all kinds of stuff, if not already broken down by your stomach acids and pepsin and everything else. What's going to happen is it's not going to get into your cells 99.9999% of the time. We're tricking our cells into believing that it's self by putting a lipid nanoparticle around it. So it goes through the lipid bilayer of the cell and it carries the payload of the RNA into, and not just the RNA, sorry, we're talking about DNA plasmids. It carries that crap that she's talking about, left over from manufacturing, into your cell. We don't want that. They said it wouldn't happen, and the 10 I forget, is it nanograms per
Grams
deciliter?
per dose.
Oh, okay, that was for regular vaccines. They came up with the standard. Okay, it's a minimum specification, but they broke their own standards. The fact is, the standard that was made was not even made for this type of product. They
Nope.
did not anticipate the LNPs carrying it directly into your cells, which couldn't otherwise happen without the lipid, without the LNP as part of this product. Sorry to interrupt. Just wanted to break it down for people.
No, I think this is one of the best podcasts I've ever done. This is synergy, dude. Um, Absolutely. And the thing is, this is kind of dark, and I'm sorry, I gotta go there. It's kind of impossible that they didn't know all this shit. We didn't need the FOIA requested pharmacokinetic data from Japan, although thank God for Byron Bridle for getting
Yeah.
that to us, which shows clearly in Wistar rats that the lipid nanoparticles in this Pfizer context traffic everywhere in the body and bioaccumulate, including into the ovaries and the adrenals, etc. There was a paper published in 2012 that demonstrated exactly this in Wistar rat, same model, same lipid nanoparticles. They use different kinds of nanoparticles, but it showed the same thing very clearly that one of the main places that these lipid nanoparticles traffic to were the ovaries. And the reason why we use Wistar rat models and mice models before we go to humans is because we're very similar biologically. That's the whole reason. So if something happens in a mouse or a rat, you gotta be careful because it might happen in a human too. Wink, wink.
I,
So another thing I want to throw in here is that there's this drug, pardon me, there's this drug called Onpatro, which is utilizing the exact same kinds of lipid nanoparticles, which act as like in the same way that chylomicrons do. It's like these things that we inherently have for fat metabolism that have proteins adsorbed, which is on the surface of the lipid nanoparticle, that traffic these guys, these lipid nanoparticles with their silencing RNA cargo to the liver via ApoE, because there are these ApoE receptors in the liver in high quantity, or they're expressed at high levels. And so these genius biotech guys have discovered, and I'm not being sarcastic, they are geniuses for doing this, but this shit shouldn't be being used in humans. this stuff traffics directly to the liver. This is all known. They've been studying lipid nanoparticles and trying to make them not toxic for frickin' 2 decades, people. But
Yep.
the thing is, that's very suspicious to me, and I have no answers to these questions so far, is how is it possible that in 2020 or whatever, whenever it was they did this, Moderna and Pfizer simultaneously solved the toxicity problem of lipid nanoparticles by coming up with this ionizable cationic lipid? What the hell is that?
It was Operation Warp Speed. Because you wave a magic wand from the executive office of the president of the United States, and everything, all the checks and balances downstream go away. And he's
Yep.
very proud of the fact that. So I'll tell you what happened today. I went to the gym, second day in a row after 4 months off, which is why my face is all blotchy and I'm
4 months,
yeah, I took
John.
4 months off. I put 10 pounds on. I was 206 pounds going in yesterday. Uh,
Oh,
I think
God.
when we met over the summer, I was probably like 192 or something. Anyway,
Well, good
so
for you for being in the gym.
I started talking. Some guy was talking about uh, getting violently ill, and the two guys were talking right next to me. So, I talked to a lot of people. I was like, what happened? It turns out he and his friend both ate a sub and they both got sick. So there's something obviously.
Subway?
What's that?
From Subway?
Oh, no, I didn't say Subway. I'm not saying any name. I'm not going to get sued by Subway. I did not say Subway. You
I'm asking
did.
because I used to eat a lot of those meatball subs, boy, and every single time.
Yeah. Oh, I got stories. I was in a hospital with 17 people. All 17 of us ate at the same pizza place.
Oh
A
God.
guy had a festering wound on his thumb, and we all ended up throwing it. I tore something in my stomach, and blood came up because it was so violent.
Oh my
Anyway, let me
goodness.
get back to the gym. I can talk a lot. So I said, well, I just want to make sure it was food poisoning. He said, oh, yeah. So I told him about what happened in Boston a long time ago. There was a wedding and everybody got sick. They traced it back to the strawberries at the wedding. And the Mexicans who sent the strawberries, they went back and traced where they come from. The Mexicans were peeing on the strawberries. That's a true story. Another one I told you about the
But
pizza.
isn't urine sterile?
What's that?
Isn't urine sterile? It must have
Well,
been handling, like hepatitis
it's
handling.
it's got a lot of nutrients for various bacteria to grow. So if
So
bacteria
is the bacterial like, sorry, I'm cutting
if
you off.
there's a seed bacteria anywhere, it's going to grow in a very nitrogen rich environment. I mean,
Gotcha.
I'm not a biologist. I don't know,
No,
but
that makes sense to
uh,
me.
so like you said, urine is sterile. But I just if you pee on something and you leave it there for a few days, there's a lot of stuff that's going to grow on that. Anyway,
Especially if it's urea rich.
that was in the Boston Globe. So if you want to research me, fact check me. I think it was about 30 years ago. I was in my 20s. I'm 61 now. So I was around 26. Anyway, so the guy in the gym, sorry for the long story that was supposed to be really short.
I love it.
So I said, the vaccine causes that. I want to gauge their reaction. They look at me, and I said, I don't mean to. I didn't even get halfway through. He said, oh no, I'm with you. And then everybody in the locker room starts like listening in. They're like, what's he talking about? So I realized that people are listening and I'm just going off, right, with all the facts that I got tons of facts. And turns out this other guy, old guy from other end, old guy around my age, 61, comes up and says, hey, what's your name again? I said, John Beaudoin. Just type it into Rumble and you'll find 150 podcast interviews.
And your computer will start smoking.
The DoD Operation Warp Speed military intelligence will come and try to suppress all of
Mm.
your social media. Um, so he says,
It's
yeah,
funny that we can joke about it. Ah, and they
yeah,
love it.
1 million
Sorry.
people died. Ah. Oh God, what a world we live in. So
And we have targets on our backs.
the guy says, um, "Do you know anything about kidney stuff?"
Oh, wow.
So he says, my brother, I told him not to take it. he took it and now he's on dialysis for the rest of his life.
And we're talking about the shots now, right?
Yeah, yeah. He took the shot. He's on dialysis for the rest of his life. Then he said, my son was supposed to be paralyzed for the rest of his life, but he's recovering and he can walk again. I said, transverse myelitis? He said, yeah, how'd you know? Well, everybody knows that. Everybody in my world, you know it. So the point is, I'm in a gym in the heart of pro-vaccine country. There is no more pro-vaccine in Massachusetts. They make it here. Moderna's headquarters is here. Their R&D is here. Pfizer's divisional headquarters for their mRNA is in Andover, Massachusetts. And I'll spare you the list of 600 companies and 47 billion in venture capital financing, MGB, and all that stuff.
Yes, please do.
So the point, this all happened out of Massachusetts. So anyway, I'm just talking in the gym, and then the other guy's telling me something else with his friend, and like it's coming out, Jess. It's coming out. I
Oh, I
mean,
know. I said this today. Everybody freaking knows
yeah,
whether or not they're the first one to instigate the conversation, like you and me. Um, Everybody knows.
yeah,
It's exactly what I said years ago. There's going to be a point, like a tipping point, probably, where they're just not going to be able to hide the bodies anymore, and that's a metaphor for everyone's going to know at least one person in close proximity. It's like, 1 degree of separation or personal experience with damage that is irrefutably because of the shots. And or I will suffice this with the measures. The whole goddamn COVID era was an attack on humanity.
absolutely. I mean, we probably don't have time to get in all how deep it goes, and I don't think either one of us really knows, and I don't think anybody knows how deep it goes. Even the people at the deepest levels probably don't even know.
No, you're absolutely right. And that's such an important point because 100%, dude, there was no predictability in this. And I know that I just got over saying that there's no way for me to believe that the regulators and manufacturers couldn't have anticipated some of the problems with the DNA, but the predictability was still there, even though we can't really predict the outcome at a population level, because you can't do that because the system is too complex. You can anticipate, the problems, but even it blows my mind that even at the individual level,
Yeah,
because we have, and this is a fact, people, we have no freaking idea from vial to vial what the hell is in there
yeah,
or what condition it's in, even if it's exactly what they say it's supposed to be. When you get that 300 microliters sucked up into that syringe, you're getting 30 micrograms of nucleoside modified mRNA in the case of Pfizer. We have no idea if that's what you're getting. We don't know how much LPS. We don't lipopolysaccharide from the bacteria.
but
We don't know
we,
about DNA. We don't know about, the condition of the lipid nanoparticles themselves. Are they amorphic? Are they clumped? We have no idea.
so
It's
what she's saying, everybody, and I'm gonna break it down to my personal experience. I'm semiconductor research, contracts and chip development. What it takes to manufacture something very complex is generally hundreds of steps of manufacturing. And there's a manufacturing quality assurance that goes on. There's manufacturing test that goes on after the product's made. Before you ship it, put it in the stream of commerce where it can hurt a lot of people. You do either full testing if it's a nuclear missile, or you do stratified random sampling of lots and so forth. And from those random samples, you can determine the safety analysis. You have cross lot variability, you have intra and inter lot variability. What she's saying is they don't
site
know. What's that? Sorry.
variability.
Absolutely. Site to site, if you meant manufacturing sites, definitely site to site. I mean, that's why TSMC can't replicate their plants in the United States. It works at Taiwan Semiconductor Manufacturing Corporation. Very difficult to move a manufacturing process when you're talking about nanoparticles, and the means at the nanometer level. But back to the point, their rules that they've set forth, they broke. And those rules aren't even for this product. It's for another product. And so you're getting a bunch of shit. You don't know what it's producing. It's producing what you want. You've got that little spike protein, but
Well,
all
we
the
don't know that, John,
well,
because
that's
of
true
frame
too.
shifting, but I'll go into that after you finish.
Okay, so you want they purportedly want the glycoprotein, which is toxic in itself, in another conversation. But you're getting all these, and this is where I need your help. You're getting all kinds of off-target proteins. They don't know what the hell they are because the code, okay, from the manufacturing with all the variability that happens, they're not testing. They don't know. So all these contaminants, it isn't just the SV40. It could be the SV40 with a tiny little change in it. And all these little changes are creating proteins running around in your body, gunking up your kidneys and gunking up your liver, because whatever is supposed to be filtering the blood has a lot of interaction with the blood. And it's all about the blood. The blood goes everywhere. Well, strokes can't be from the vaccine, and kidneys too. I mean, it's two different things. It's like, where does your blood go? Where does your blood go? It goes everywhere. The whole skin problem. I got to show you a graph before we get off. I'll show you. I did it last night. Unbelievable. So if you could comment on variability, what it's actually producing. Am I wrong in saying there's all kinds of off-target proteins that are messing
Oh,
up?
you're 100% right. Like if you guys still believe in the peer review process and nature and science, which I'm sorry, I still do. I'm sorry to everyone. I have to believe in something, I think. A paper came out in Nature recently that was talking about this subject matter called frame shifting. Here's biochemistry translation 101, okay? The design of these products was based on manufacturing a strand of messenger RNA, which is nucleoside modified, that would be introduced to your cells via these lipid nanoparticles and subsequently translated by your ribosomes, which are these little machines that take messenger RNA from your cells and translate them into necessary proteins. When things are coded properly and read properly and at the right rate, you have this mRNA. Think of it like a shoelace being pulled through a burger, okay? I always do this. And the forward end is like the shoelace being pulled somehow through the burger, and for some reason the burger reads the shoelace in sets of three, what we call bases, which are also subsequently called codons. So triplets called codons. At the outside end, the butt end, you get a translated amino acid from this codon. So like an ATT will be translated into a whatever that is. I don't remember. Look up your codon table. And those amino acids will form a string at the butt end of the ribosome, and necessarily based on their physicochemical properties and forces and stuff, they will fold into a three-dimensional shape. That's very, 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 very important. Protein folding conformation is more important than most people talk about. If you have a protein that's misfolded properly, especially an essential protein, that can lead to all sorts of things like prion diseases, and those are way worse than viruses. So it was actually because of the N1 methyl pseudouridine replacement of not one, not 10, but 801 of the uridines in the spike code. Every single one was replaced. That it turned out that instead of this shoelace being pulled by the burger at a specific rate to translate into this spike protein, there was slippage, something called slippage, because there were sometimes three N1 methyl pseudouridines stacked in a row. So it would slip. And what that would mean is instead of like a set of three, a codon being read, to translate into an amino acid properly, it was slipping and it was starting to read, say, the shoestring sentence at the wrong starting point. So what was coming out the butt end wasn't what was the target protein, the spike protein. It was something, who knows? We have no idea.
Yeah.
It cannot be characterized because we don't have a submarine that can go into cells and tell us with cameras and shit. So we have all these aberrant, potentially aberrant off-target proteins that are misfolded weirdly. And remember what I just said. If you have misfolded proteins, a whole whack of shit can go wrong, including cryogenic diseases. This is a fact. I don't need it to have
Well,
been published in Nature to believe this. This is just one of the predictable things that the manufacturers should have predicted during the R&D of these products.
let me add a little bit, if you don't mind. There's a lot of engineers out there. I'm an engineer, right? Not a very good one, but I'm an engineer. So we take classes in chemistry, physics, and then we move on to mechanics, statics, and dynamics, I think they call them. There's one called material properties. If you take a class of material properties, you learn about the lattice structure of, say, diamonds versus coal. You get carbon, but carbon can be in different forms at the molecular level. What Jessica's talking about is assembling various parts of molecules and molecules into these long chain molecules. And these long chains, or of whatever material, biological material, and when you create these long chains, they can kink up. Now, a rubber band.
That kinking is another thing. Sorry.
Yep, a rubber band, you can take a rubber band, right? Rubber bands are called, they're polymeric, all right? They're polymers, and they kink up. And they're in their natural form, they're all kinked up. But when you pull a rubber band, everybody knows that you get to a point where you can't pull it anymore. You've pulled all those long chain molecules into their linearized form, and you can't pull it anymore because that's it. You've reached the end. If you create all these proteins, and these things are, and even when they call it a short chain protein, is still a long chain molecule. All these things in your body, they can adhere to each other and create. You've heard about these amyloid clots, these white clots. You've got all kinds of stuff going on in the body where if you change the formation of molecules, large, and am I right in saying molecules, or should I say?
Yeah, I just call them molecules. It's
Okay,
fine.
they're
They
really
are
long.
molecules.
They're wicked long, right?
Polypeptides, folded
Okay,
polypeptides, whatever.
you're manufacturing this really long thing, and it can get kinked up, it can fold back on itself, it does all these things that the material properties of the various atoms within it do. There's charge. Charge has to do with whether or not it's going to have an affinity for something else, right? If a positive attracts negative, attracts positive, like the stacking of poker chips of the erythrocytes with the rouleau formation. You've introduced a charge. The only way that happens is you've introduced the charge to the erythrocytes such that you have a north-south, and they stack. And then people have trouble breathing because you've reduced the surface area of the erythrocytes because you just took off half of, or more than half of all of them because you stacked them. And I'm off target here, but what I wanted to say is with the long chain molecules that she's talking about, that these are being assembled by the machinery that's put in through the mRNA. It's bad. It's producing all this plaque or this junk, and they don't even know what it is. And it's really bad. And now you can understand basically what Jessica's saying. I hope I cleared that up, or maybe I made it more
Well,
complicated.
there's another facet, and I'm glad you brought this up because off-target protein production problem is a thing, but it might actually, I know this is going to sound weird, but it might be worse if you have on-target protein production because of amyloids.
Mm.
I've written a number of articles that demonstrate, and these are published in ACS Nano, that there are amyloidogenic peptides. These are strings of amino acids that are amyloidogenic, which means that they form amyloid betas sheets. They stack up on top of each other, and they're not degradable, like at all, by your body. So if you have amyloids formed, they don't get broken down, and they tend to get involved in the clotting pathway. When you have platelets and fibrin called to action to clot blood, those amyloids can get involved, and those can form really dense clots that are really hard to break down.
Mm.
So there are amyloidogenic peptides in the frickin' spike protein. This is, besides the fingerprint of the PRAR, the furin cleavage site, this is one of the things that leads me to believe the most that this was made to do harm.
Yeah.
Because I can't imagine why the hell or how the hell it would be possible for anybody doing the in silico part of designing the DNA for the spike to be used in an injectable product, not to have run that through bioinformatics software to look for amyloids or to look for molecular mimics.
Yeah. And when she says amyloidogenic, anytime you hear gen, you're making something, whether it's carcinogenic, making cancer, amyloidogenic, you're birthing something. So it's creating amyloids, is what she's saying.
Yeah. And I think this really explains a lot of what we're seeing in terms of both micro and macro clots. You mentioned the rouleau formation. I have a very good friend who's a hematologist, and he said rouleaus aren't actually that weird. Just so that people know, the spike protein itself, if it's actually translated properly into this, pre-fusion trimer thing, it's negatively charged. By the way, the lipid nanoparticles are also they have a net positive charge. Spike is positively. Red blood cells are negatively charged by nature. This is why they repel each other and they keep their distance while, rushing maddeningly fast through the circulation. It's why they don't stick together. So if you bring something that is able to interfere with what's called the zeta potential, which is this repulsive force between red blood cells that is positively charged in a certain quantity, you might experience the effect of red blood cells coming together because that repulsive force is being acted on by something that interferes with that. So it brings them together. So anytime you see rouleau formation, it's like, you guys remember those candies when we were in the 80s and they were called Rolos, or like butterscotch candies?
Oh yeah, yeah. The Rolos. I remember Rolos.
Lifesavers.
Yeah.
So yeah, it looks like that. The reason that happens is something happens to the negative charge that's inherent in these red blood cells to stay apart. The most likely villain here is something positively charged, and it's suspicious that the spike protein and the lipid nanoparticles are positively charged, especially the LNPs, because they have these cationic lipids.
Let me jump in because I'm going to propose something to you, and you can tell me I'm full of crap if you want. I have no ego in this at all. So, it's hard for me to believe that many spike proteins can touch that many red blood cells. So I go back to the formation of the red blood cells to the hematopoietic stem cells. If you mess with the hematopoietic stem cells and how they're formed, and they for the audience, that's the base level cell for your blood that gets differentiated into erythrocytes, which are reds, all kinds of white cells, neutrophils, eosinophils, and everything else. And then also the platelets, which are called thrombocytes. So if you dysregulate the blood formation in the marrow through the spike, maybe not the spike protein, maybe the mRNA gets into some of the machinery of creating blood cells and creates some off-target blood cells. I don't know, but maybe you have different charges such that the reds are clumping together, the thrombocytes are producing too much fibrin with no off switch, and then you've got the white cells all messed up, attacking self. Am I wrong about the possibility of hematopoietic?
Oh, there are also papers that show that molecular mimicry is a problem in the context of, the bone marrow. So there are, like, I don't know about access, but it's an interesting thing to follow up because we don't know if and how, the bone marrow is being affected, or the stem cells or the germ cells or
It's
whatever.
one of the strongest one of the strongest cancer signals is bone and bone marrow cancer.
I
I don't know
know.
if you know
So
that. I don't know.
that's exactly right. So, we need to, this is why talking about this shit that affects literally every single person on the planet is so important, because we might actually be looking at an existential crisis, and first of all, we need to acknowledge it,
Yeah.
that there's a problem. One of the things about cancers is that sometimes, most of the time, they don't show up right away. It's going to be real hard to trace back all of these emergent cancers that we're seeing now, 3 years out from, the first shots that were doled out. It's going to be hard for us to link those back to those shots, but I have no doubt in my mind that they were a driving force behind it. If not the cause, the primary cause, they're absolutely the driving force. There's something, the SV40 is insidious in the world of cancer because it can bind P53, which is the guardian of the genome, this tumor suppressor gene. It can induce C-gas sting, which is an inflammatory pathway that can lead to cancer. It can help other DNA fragments get to the nucleus of cells for genomic integration. I mean, god damn, there are, pardon my language, but there's so many. That's just one avenue too. I mean, it's like I'm always, what Kevin and I talk a lot, and it's like every time we turn over a rock,
Yeah.
there's something else. There's some other critter that we didn't expect to see crawling around under there, and it has a lot of legs. It's like, wow. And we're going, circling back to how we started. I mean, God, like how is it possible that we're in a milieu of censorship when we're talking about this kind of subject matter? We're talking about the genomic health and fidelity of a species. Really, that's what we're
Uh,
talking about.
we're the species she's talking about.
Yeah, yeah, yeah. And not only us, they're starting to inject animals now. So like these are the
Yeah.
animals that we're going to be
With
consuming. They're
with the
not going
self,
to tell us.
you're right. It's messing up my food supply, and I want to, but also they're going to be injecting them with the self-amplifying stuff so that it
They already are.
sheds
I
to
can
human.
guarantee you, John, that the vets who are using the new rabies shots in the States, they have no freaking idea that it's
And,
self-amplifying.
and so what that means, it's your pet is shedding the vaccine onto you as a human. You're now
But
taking a rabies vaccine intended for the dog. You inject the dog, and it goes to the human companion. That's how messed up this is. People
but
don't
it's
get.
more messed up than that because the self-amplifying RNA carries a gene called RDRP, the RNA-dependent RNA polymerase, as part of an alpha virus, the Venezuelan equine encephalitis virus backbone. They picked it for some reason as the prototype
To
that
kill
contains.
people, to kill us all. That's why they picked it.
Well, it has this photocopying gene, which is this RDRP, and they took out the guts so that it's not replication competent. And they swapped in spike genes, and in another trial, they swapped in flu genes, the neuraminidase and the hemagglutinin genes. So what that means is that instead of having a finite number of mRNA copies for your ribosomes to translate. What this means with this photocopying gene is that it can reproduce the template over and over and over again to exhaustion, who knows till when, because none of those studies have been done, to produce massive more quantities of whatever
Whatever.
gene is encoded, whether it's spike, whether it's flu genes, whatever.
Or
And
the off-target crap that they don't even know what they're doing.
exactly. So there's so little. It's just like if people understood how freaking little they knew about how much genetic material was actually and the nature of it being brought to cells and how much protein and what proteins were being produced and which cells this was happening in, and the fact that the self-amplifying thing is touted as being, so much better than the mRNA because we don't need as high of a dose, and without even considering the fact that there's no such thing as dose.
Right.
I'm making a slide deck right now to really like show this to the layman, like why this is a ridiculous thing to compare, first of all, infection to injection, and why dose is like some kind of myth with regard to these things, because they're claiming that a dose for Pfizer, for example, is 30 micrograms of RNA material. But first of all, that's not all you're getting.
That,
Second of all, that means nothing when you talk about how much protein is going to subsequently be produced, because we don't know which cells are transfected by LNPs. We don't know how much coding material is brought to those LNPs, if it's 5 transcripts or 100. We don't know how many times those transcripts are read, and we don't know what proteins are being produced or how much once they're read.
So let me break this down again. It's not a vaccine. It is a vaccine manufacturing production.
Prodrug.
It goes in your body and it produces the vaccine in your own cells, if you want to call it a vaccine. It produces the
It
antigen or whatever.
produces the foreign protein.
But there's no off switch.
No,
Well, it'll stop after a while. So
it'll stop when the cell gets killed.
yeah, when with regard to dose, you can get a dose of penicillin, right? And you know how much penicillin you're getting. That penicillin isn't going into your cells and creating more penicillin. It's not setting up shop it's not a manufacturing plant. You don't talk about a manufacturing plant as to how many products they make. You talk about it, how many products do they make over time without the derivative of time. Okay, there's nothing dose related about this product. It's not a dose, it's a dose over time. You're creating the antigens in your cells over time, and without that time variable, there's no point in talking about dose. It's ridiculous.
Now, dose is related to half-life. So, like you'll take a certain amount, and drugs and biologicals are completely different, and
Well,
vaccines
the
are.
limits to the dose that you say the dose limit on a product is related to half-life.
Yes.
But the word dose is okay, just to be clear.
True. It's hard for me to believe at this point that this isn't just another way to make people feel better about something that's potentially going to hurt them. It's the same thing as calling these things vaccines. They're not. They're calling them vaccines under the guise that they induce some kind of frickin' antibody response, which is stupid. That's too narrow, and it's already been disproven. I mean, these things actually induce tolerizing environments. We know that now. It's in the peer-reviewed literature. They make you more susceptible, which is negative efficacy to SARS. The number one reported adverse event in VAERS, the pharmacovigilance database in the US, for years and remains so, is COVID-19. This
Yeah.
is the vaccine adverse event reporting system in the context of the COVID-19 shots, which
Let,
primarily were the Pfizer and Moderna shots in the US.
let's not forget about shingles and RSV and everything else because it crashes your immune system and now you're susceptible to everything else. It's not a vaccine, but the pandemic, Jessica, I say the pandemic is hubris, not virus. You've got
Yep.
people with so much pride that they think they can make a product that's going to save humanity, and
But the other thing about that is I don't like, a lot of people are going around saying, oh, they're trying to kill everyone, but I still don't believe that. I think that they just don't give a sorry, a
well,
shit if people die. And
this
there's
is
a big
where
difference between that.
yes, and this is where I always jump on pronouns. So when you say they, there's different mindsets for everybody. People don't know what I do. They don't know what to call me. Like I'm an engineer. I got an MBA. I got some, finance stuff and some economics and some engineering for the
You're
physical.
John.
Yeah. I am a whole systems analyst. What does that mean? Most of everything that happened has to do with behavioral modification, psychology. And they knew a lot of stuff they were doing, especially with the CARES Act and everything else. They modified the behavior of hospital administrators, doctors, policemen, firemen. They modified the behaviors of people through these policies, and that is how you modify
And
behavior.
through incentivization
Ex-
programs.
exactly.
Let's pay people every time they write COVID-19 on a death certificate. You know that better than anyone.
Yep, yep, I do. I've got so many presentations about. I can tell you the dates that they remdesivir, by the way, in the video you did with Kevin, with the TFTC, TFTC.
TFTC. Whoa! Go Marty.
I got it. TFTC.
Everybody go watch Marty Bent.
So I
TFTC,
saw that was
TFTC
great.
21.
I recommend everybody watch that. I mean, it's 2 hours, but I'm
No,
back in.
it'll fly by, dudes. Like seriously, I
It
usually
is.
don't get into watching myself because I'm like, oh my God, you sound so stupid. But it's really good
It's
because
very good. Very
Kevin
good. I
and I have
recommend.
good synergy because he knows stuff that I don't know, and I know stuff that he doesn't know, and Marty's like kind of like sitting there like Like, oh my God, what are you guys talking about? No, I'm kidding. He knows a shit ton about stuff that I don't know too. But when I say he sits there like this, I kind of mean it from he's kind of shocked. He's very normal,
Don't worry
beautiful
about him.
human
The
being.
information that you and Kevin, I don't care about him. Sorry. I mean, I
I
hope
do.
if I
He's
talk to him,
brilliant.
I'll care about him when I talk to him. My point, though, I want to get back to my point. Watch the video. That's the point. Because you do get into these behavior modifications. You do talk about the incentives. And I'll tell you, November 2nd, 2020 is a date that killed a lot of people. When they put a bounty on people's heads in hospitals, you got the hospitals, well, we heard about this remdesivir, well, maybe we shouldn't use it, maybe we should. Some hospitals used it, some didn't. They didn't use it en masse. But when they said, if you use remdesivir, you'll get a 20% adder for the entire hospital bill. You get them in the ICU, it's $1 million to the bill. You get an extra $200,000. I don't care about $300 a dose. I don't give a shit about excuse my language, about $300 a dose. You get $200,000 just for running that stuff through somebody's veins on a positive, crappy PCR test. That's it. And they killed people and they murdered them based on this. And the doctors looked the other way. So when you say they, I'm going to get back to where we started this thread. It's different because the hospital administrators were solicited by the money. They were going to lose a lot of money. Nobody was going to the hospital in 2020. So they did that so they could make their numbers. The doctors did it, some of them because they believed in it. They believe the literature. Some of them because they don't want to know.
Peer
So
pressure,
they look the other
like
way.
silly teenagers.
Yep, some knew it and they didn't want to lose their jobs. So they were coerced by the board, the board certifications, the certification boards, also by the hospitals. Do this or you're going to be fired. They didn't want to lose their jobs. And they made examples out of people. It's all about behavior modification. There were laws and procedures.
Say something though, if you want to hear what I think.
Yes,
Um,
please.
that whole thing about I don't want to lose my job, I get that for tradies and people like that. Not for medical doctors and physicians and nurses, because I hold you guys in the highest position of all. I don't think that there's
Doc,
no,
no,
seriously.
no, no,
No,
no.
let me
That's a
finish. A
fallacy in
medical
society.
doctor
Yeah,
who actually is well trained and who is like free thinking and who maybe learns surgical practices is somebody who has essential and unique skills that other people don't have.
yes.
I'm sorry, I'm going to say it. I think it makes them more important than other people. Like because if I'm like crashed at the bottom of a ravine with broken bones, I'm gonna want the best freaking medical doctor at my service. I'm not gonna want a philosopher. I'm sorry. I know that sounds bad,
No, I
but I'm
agree.
just trying to say that medical doctors, besides the fact that they swear an oath to do no harm, they have a role that is a role that other people don't have, not just because of certification and oaths. They have a role to play to protect, not only to safeguard and heal, but to protect people, not just their patients, but people in general. I know that I sound like, a naive granola, but
No,
that's what I believe.
no,
And so
let me break
I,
it down.
I don't give them a pass at all. I
Let
think
me,
every single medical doctor who has the appropriate training and licensing, who frickin' had any inkling that what they were doing was fucking murder, for example, pardon my language, and kept doing it, are they
I
should
go.
be like complete. They should lose their licenses, not the people who are promoting hydroxychloroquine.
They're actually criminals. It's more than
Yes.
just losing your license. So let me break it down for legal, moral. We got to separate legal and moral.
Sorry, I
Uh,
got a little passionate there.
no, no, I think it was great. It was great because I want to address this too. People make the mistake that where a doctor has that special skill you mentioned and can save a life, they make the mistake in believing that person is more ethical or moral. It's got nothing to do with it. Doctors are some of the biggest psychopaths that ever lived.
Now,
No, all through history, Jessica, all through history. You know,
Oh, let me believe. Let me believe.
I mean,
It's almost Christmas. Come on.
no, give you an example. There's a guy, I think it was in Natick, Mass, or close to Natick, Massachusetts. This just goes back probably 30 years, maybe 25, 30 years. His child died with organ failure at whatever age, had trouble, 2, 3, 4 years old, finally died at, I don't know, 4 or 5 years old. Um, Kidney problems, heart problems. So the wife was found murdered in the home, but her organs were missing and they were stuck on stakes in the backyard. Her kidneys and her heart, whatever was wrong with the kid who died, that's what that doctor did to his wife and cut her organs out and put them on the stakes in the backyard. Another doctor murdered his wife in Massachusetts. I'll stop now, but my point is, they're
Yeah, I
people.
think you're talking about outliers there. They sound
No,
very
well, yeah,
psychopathic.
of course, those are outliers, but doctors are no more moral or ethical than anybody else. They have a code that they're supposed to live by. They only do it not because it's the right thing to do, but for fear of repercussions, say from the medical board. The what you mentioned, where they have that special skill and they have to do it, they have a legal duty to act. You and I, if we come across a person
We're
drowning,
not allowed, yeah.
well, there's the Samaritan laws are different by state. In some states, once you begin trying to help, you have to finish helping. You can't stop in the middle and say, "Oh,
But
I'm a."
I thought if you didn't have like technical training and like tried to save someone's life, for example, and you died, you could actually go to prison.
No, it's dicey. There was no intent. You can't go to prison unless they can prove that you intended to harm the person. If you're trying to help and kill them, you could be sued for wrongful death. So
Right.
it becomes a civil tort, not a criminal thing. It's yeah, we don't want to go there. It's very, very complicated. But the point is, doctors are not any more moral or ethical than anybody
But
else.
they're supposed to be. That's my
No,
point.
that's the misconception
I
that
know.
somebody
Well,
has about doctors.
this is the sadness that we're facing these days. I mean, I'm going to be the first one to agree with anybody that says that med school now is just it's a monkey taking a multiple choice test. I mean, it's just like school is. It's not about creative thinking. It's not about, maybe some schools are, but most medical schools are just there to indoctrinate. Here, you have to memorize all this shit, and if you don't, it's an impossible schedule. It puts stress on the mind, on the body, puts you in huge debt. If you make it through, you have to clear up this debt, which I guess is the first task of anybody who got through med school. Maybe that's where the incentivization comes from for, selling yourself out, selling your soul. But I'd like to think, I really, really, I need to hold on to hope here that there are still some young, eager, curious, thriving minds out there that did go through med school who didn't get, like, I went through school, like, for sure it was the 80s and the 90s, and I've been in academia all my life, and it hasn't affected me. I've always been a black sheep, if you want to call it that. I've always been on the outside. I've always been made fun of. I've always been the underdog. I've always been the one winning awards, by the way. But it's like there are people, and I think,
Everybody's an individual, Jessica. So yeah, there. I have a great
and
doctor.
I believe that there are enough.
I I don't think I have a chapter in my book, which is right here. I have a chapter in that book. It's called EBM and Dr. Backer. And in it, I explain what happened with your country did this.
Yeah,
The,
my country is like, I
the,
don't know, man.
the Canadian Healthcare Act of the mid 1960s funneled money for research into especially McMaster University. And they came up with a lot of research papers that were disseminated and agreed upon. And they came up with what was coined maybe 15 years later called evidence-based medicine. And that was in the early 80s. And these two guys got really famous, and evidence-based medicine became this thing. By 1992, every North American, United States too, every North American medical school taught EBM which means you do what the central authority says. You follow the literature, follow the literature, follow the literature. So that means if there was a study done halfway around the world in India with different genetics, different water supply, different foods, different minerals in the area, you follow that study. You don't look at the patient standing in front of you who is completely different genetics, different foods, different water. And you know him because you know, as a doctor, you know the local area. You know that, oh, he's Portuguese. He can't eat this kind of fish. Forget about all that. Do what they did in India because there's a literature study that says that. That's what's happened to medicine since 1992. And if you look back and you ask doctors what happened, let me break it down real fast. They could not have done COVID to us 10 years earlier because those doctors hadn't retired yet. It's the only, they had to wait, or they didn't wait because it wasn't conscious. It was serendipitous that COVID actually worked. But the reason it worked in the psyche of the doctors that were in, and the younger doctors are in the ERs because it's a real grind. The reason why it worked is because all those doctors went through this new evidence-based medicine in all the med schools. That's why we got all screwed up. Sorry.
That's no, don't apologize. That's brilliant. So that answers a lot of questions I have about the CDC's power and the FDA's power, because my impression is that the world kind of takes the lead of the CDC recommendations, which come from the FDA, basically. I might be wrong about that, but I think that's the sequence of events. But
Um,
what you just said,
don't forget the NIH. Sorry, just
yeah, right, of
the
course.
NIH is 6 times bigger than the CDC in budget.
Right, but what you just said is wow. It's kind of like the way that med school was infiltrated in a very specific way. Um,
Centralization. It's centralization of authority and power into the now CDC is the recommending arm, FDA is the approving, but NIH comes up with the protocols. Okay.
exactly. So, but there's no way. You're absolutely right about the age group. Like, there's no way for anybody who just got out of med school recently to, like, if you ask them anything about this, they probably would have no clue.
They don't.
It's
They don't
just
have any idea.
it's the world that they live in that's considered normal. Like, this
Yep.
is how we do it without even having the training. But this is the
It's
part that bugs me, but I
it's
can't
around
judge because I am who I am and I'm nobody else.
it's
And
around
I'm
55.
lucky.
Sorry, it's around 55 years old. That's how many. It's around 55 years old because I mean, if I'd gone right into med school out of college, I would have graduated in 1990. So and I'm 61. So 59, I guess, would be the cutoff. But anybody under 55 is EBM and they don't know any better. They don't know any different. Talk to Richard Amerling. Dr. Richard Amerling.
Yeah,
I,
me too.
I think he was the president of American Physicians and Surgeons, American Association of Physicians and Surgeons. Great guy. And I agree with him. He's older than I am, so I'm not going to say he agrees with me. I agree with him. We have made and come at it from, I didn't know him. And we said the same things and then kind of found each other. And he's a
From
doctor.
different disciplines.
He's a doctor. And he said the same thing. Evidence-based medicine has ruined medicine. They're all centralized. And I'm like, he's giving my speech. And I didn't even know him when I made the speech. So I've corroborated it in a real guy, and he's great. So
So, everybody, one of the take home messages of this podcast is go look up what evidence based medicine is and how it is very implicated in everything that we're all still going through. For the people who don't understand why we're still talking about this, it's because it was the dry run, okay?
yeah,
Trust me.
yeah. I've got a short story called it's called A Short Story About Bill, I think. Bill's a doctor and he meets Frank and as they went to med school together, it's like, how's your life? How's your life? And Bill's like, well, I had some trouble. I couldn't sleep. I was going to get a divorce. My kids hated me. Oh, what happened? Well, I had to make a decision how to treat a patient. And the literature said to do A. And I knew that B was better, but I did A. And I knew that it would create a 5% more chance of dying. You had a 95% chance of living if I did what the literature says, the CDC, right? or I could do what I wanted to do, which was 1% chance of death or a 99% chance of living. And I chose to do what the literature said, and the patient lived. But my psychologist told me that I felt so guilty that I was angry at my kids, my family. And then Frank says, "Oh, I got the same story, except I did the 1%. I did the right thing, and my patient died." And so they took my license to practice, and now I'm selling pharmaceuticals. But it's a short story that shows, and I do an analysis. That difference in the 1% and 4% difference of the doctor knowing the patient, doing the right thing for that one patient, rather than doing what the literature says. If it's a 4% difference, you multiply that across society and you're murdering 20,000 extra people a year by that particular diagnosis. And that's what happened. That is what happened with all those other drugs. And we could start naming drugs, right? They're like, oh, they killed people with that drug. But the COVID vaccine was the coup de grace. It's like, wow, they really killed 1 million people in the
Well,
United
it's ongoing, right? Like the after effects. I dare say, I don't want to be a conspiracy theorist or anything, but I'm starting to see a lot of this flu hoopla all worldwide, and I'm starting to wonder if instead of chikungunya, they're going to use this flu as the way to symptom launder all the damages and deaths like happening from the COVID shots now.
I'm afraid to tell you what I think because it's not good.
Why? No, go ahead. Everybody's on edge now. Let's let's keep them on that edge and hold them back.
I do have a good part. I'll tell you after, but they're going to do the flu thing like you said.
No, they're doing it. I can
They're
see it.
doing it right. What if they release something to kill a lot of people who
Well,
don't?
they could do that too. Like, I'm sorry, but I still have unanswered questions about where the fucking flu went. Sorry, where the flu went in 2020, right?
Well, it didn't. I mean, we had the flu. I could go through the numbers with you if you
Yeah,
want.
that's just a data thing, in my opinion. But
Yeah.
on the other hand, it kind of makes you wonder after learning about everything that they're doing with gain of functioning and releasing of this and that, whether or not they're monkeying around with flu viruses and like just sporadically releasing them.
Yeah.
I don't know, it's a stupid idea, but who freaking knows what's true these days?
Well, here's the good part that saves kind of the narrative. I believe in God now after my son died. I've seen so much evil in the world. There has to be a balance. And so I believe in God. I know that the virulence and transmissibility oppose each other.
Hmm.
And since the turn of the century, there's been one thing, the Spanish flu, that killed a lot of people. And you can argue what really killed people. Was it
But
overdose?
that was a synergistic relationship between a virus and a bacteria. That was that.
Exactly.
Oh,
I,
good, you know that.
yeah. Well, there's a bunch of stuff. I mean, I'm old enough to know my older relatives who lived through it and talked about it. When I was very young, my the great aunts and uncles would talk about funeral processions down the end of the road from St. James Church to St. James Cemetery was at the end of Mulberry Street, and some other streets that they lived on in Haverhill, Massachusetts. And they would talk about 3, 4, 5 funerals a day, every day for months, every day. And people would turn black. Like, why would they turn black? That doesn't make any sense. That's like,
Like
so what's that?
that sounds more like the plague.
Yeah, but if you think about it, the aspirin thing really dwells in my mind quite a bit. They didn't have a dose limit for aspirin, and people started taking aspirin back then, and they're like, "Well, more is better," and they're bleeding a lot, and their skin showed it, and
Oh,
that's one thing. The secondary bacterial infection is how most people, I believe, died from the
yep.
bacterial pneumonia, especially because they were wearing masks and rags on their faces and they were rebreathing all that shit. But the reason I bring it up, I keep on getting off tangents. There has not been a pandemic, and the word pandemic recently invented. It cannot be. There can't be a pandemic because if something's so bad it's going to kill you, it's going to blow itself out quickly in a regional area.
Like
They,
Ebola.
yeah. And they say, well, we get on planes now. It's like, if it's that bad, you're going to know before you land. If you're going from Japan to here, Africa, if you cross an ocean on a plane, that plane is going to be quarantined. That guy is not going to. So it's all bullshit. Pandemics can't happen in the manner in which the government purports pandemics to happen. It can't happen. The other thing, Jessica, is I looked through all the movies and TV shows. Have you seen my slide on that?
Which one?
The movies and TV shows? So
I don't think
I have two
so.
slides. I have two slides. Yeah, let me see the cat. I'm not a cat guy, but I can see the cat. In the last 60 years, there were 60 movies and 60 TV shows about pandemics.
Yeah, yeah.
Society's
I'm
been programmed to believe that this crap can happen. It can't happen in the manner in which they purport it can happen. That's it. I'll stop now.
No, you're right. And this is a very important point that Marty and me and Kevin touched on. If you have a really virulent virus, like something really deadly where you bleed out of your eyeballs, like Marburg and Ebola, unless you're like right there, it's not going to affect you. And by the way, hemorrhagic fevers are kind of like merciful in a way. I've done a lot of reading about this, and it does something to your brain before you die. So I don't even think you're aware that you're dissolving. So it's kind of like, I don't know, it's a silver lining if you ever,
Oh, it is. It
if
is.
you ever melt from a virus.
It makes sense. Hey, I want to get to myocarditis and acute renal failure.
Okay, but people are going to get bored because we've been talking for an hour and 35 minutes. And Lammy's starting to give me those signs that
All
he's
right. All right.
tired.
It
He's
just
tired of his mother talking to this man.
So I'll go real quick then. The myocarditis thing, your paper, what's the status with the
Right.
McCullough? What's the status with the McCullough paper?
Oh, well, you should know this.
I
It's
know.
It's
Is
been
it
since published under a new title, Determinants of Something Something, in the Journal of Therapeutic Drug Therapy. I don't remember. In 2023, we got that republished.
okay?
I sent it off to Peter. He's the cardiologist to like go over everything. He
Yeah.
brought Nick Pulcher on as a third author, and we got that through peer review in Therapeutic
Okay.
Advances in Drugs or whatever it's called. And yeah, so the main nitty gritty of that original myocarditis paper has been published. But Bick and the Pub Smear crew, they're going for that again, that paper again. I was
Did,
actually
did you have the guy? Did you have the two teens? Did you reference that paper? The two teens who died of myocarditis? That was James Gill. Did you reference a Dr. James Gill?
I'm
No.
sorry that I can't even remember.
That's okay. No, no, don't worry. Don't worry. I was just going to say I have the death record. It's Michael Williamson in Connecticut. He was 16 years old. And that was a pretty famous paper everybody talks about. So I have his death record. Lastly, let's see. I want to make sure you understand where I am on myocarditis and other people. When I say it's 150 times more of the in the death records, acute kidney injury, I'm not saying that acute kidney injury is 150 times more than myocarditis. I think myocarditis is huge. It just doesn't show in the deaths. I think a lot of old people who died, it's not just young males. It's not. Old people are dying of myocarditis, and it's being called cardiac arrhythmia, cardiac arrest. It's being called all kinds of things, just an end STEMI heart attack. They don't know. They're not doing the autopsy. So it's huge. So
Oh, I know. And actually, the reports aren't really updated in VAERS. So if you get a preliminary report filed and get a permanent VAERS ID for myocarditis as the primary AE, and that person dies, and even if you report that,
yes.
and that ID ain't going to be updated. So you're not going to see as many myocarditis associated deaths as you would. However, having said that, I recently generated, was it myocarditis, I think? No, it was COVID associated deaths. But anyway, there are reports that made it through. So that's it's like a signal within the signal. Like it's a tip of the iceberg, if you know
Yeah.
what I'm saying.
They're not testing for it. They're not cutting bodies open to put the
No. They could be.
if they did, they'd find myocarditis in probably 30 to 40% of all these things. So that just so the audience isn't mistaken, I'm not saying that AKI is 150 times more. I'm just saying it shows in the data, and it's the low-hanging fruit to do investigation. And what needs to be done is not a bunch of more research papers. What needs to be done is getting on the government's ass to say, we know you're hiding the data on purpose. You could solve this in 2 friggin' weeks. That's it. 2 weeks, and we're done. all the debate and all the society ripping itself
Yeah, but they're
apart.
never ever going to like unsilo that data. It's
Yeah,
never going to happen. Not even the people within the CDC. I know this now. They have access to the data. Like you wouldn't believe how
we can talk.
siloed
We can
this
talk
health.
offline. I can't talk to you about that, but I'll tell you
Oh,
something
okay,
offline. But you're right. I'm saying you're right, and I can give you an
I
example, but I
know.
can't talk about it. So, what you want to close this up soon, and you probably
Yeah,
want
I
to the
actually have
day.
to run because
Okay.
I will have to talk on the phone. But yeah, I have to run because I have something at 5
All
to
right. Well, I really appreciate your time. Hope that the audience got something out of it. So that should be good. Thank you. Really appreciate it.
Awesome. I meant it when I said this was like pure synergy, awesome podcast. Yeah,
We
it's a good one. And people are gonna watch this, trust me.
thank you. Really
You're
appreciate it.
welcome. It was really fun and let's get together with Kevin soon, okay?
Yeah, no, that'd be great. That's a great idea. All right. Take care, Jess. Have a good one.
Bye, everybody.
Bye bye. Okay, everybody. I said I was going to show something. I'll show it really fast. This is 21. The red line is the beginning of 21, January. So July is right there. That's July. And that's January. So 6 months after the shots were introduced, you see this crazy, crazy increase. So that's just a bonus for the future. See if I could get some kind of published thing out of that. Have a good one, everybody. I appreciate your time. Again, thanks. Last Boomer Podcast signing out. God bless you. It's all about truth. Just want truth. That's it. All right. Bye.